This is Chapter 9, Part 3. We've been talking about some of the biochemical pathways by which cells harvest energy, which essentially means converting sugar, typically glucose, into molecules of ATP. And in the last section, I walked you through the 10 steps of glycolysis, which will convert glucose into 2 molecules of pyruvate. This is happening in the cytoplasm outside the mitochondria. We also produce a couple of ATP and a couple of reduced electron carriers, NADH, in the process. That pyruvate then gets imported into the matrix of the mitochondria, where it's oxidized in this pyruvate oxidation step. That converts the pyruvate into a molecule called acetyl-CoA and produces another reduced electron carrier, NADH, in the process. That acetyl-CoA then feeds into the citric acid cycle, which consists of another 8 enzyme-catalyzed reactions. The citric acid cycle is also taking place in the matrix of the mitochondria, and for each turn of the citric acid cycle, we're making 1 ATP, 3 NADH, and 1 FADH, which, as we discussed, is another electron carrier. And so overall, per glucose that we started with, we can see we've really only made 4 ATP so far. And that's taking into account 2 turns of the citric acid cycle. So if the whole goal of this exercise is to make ATP from glucose, we're really not doing that hot. And you might remember I told you that you should be able to get up to 32 ATP out of that glucose molecule. So what we want to talk about now is, well, how are we going to make the rest of that ATP? And what I want you to take away looking at this figure right now is there's still a lot of energy on the table here that's been stored in these reduced electron carriers. So NADH and FADH molecules are still carrying energy. And so what we're going to see now is basically using some of that stored potential energy to generate some of those additional ATP. And again, we have a bit of a metabolic fork in the road. You might remember we discussed if you have oxygen present, right, we're going through these steps that we've discussed, the citric acid cycle, and this process called oxidative phosphorylation. If not, you carry out fermentation. And we'll talk about both of these before we get to the end of the chapter. And so this term oxidative phosphorylation sounds complicated, but bottom line is what that means is making ATP in the presence of oxygen. And there's really two parts to this process. There's the electron transport chain, and then there's this phenomenon called chemiosmosis. And those two things are coupled together. We really need both of them. We need oxygen to be able to generate this ATP. And I'll show you what this looks like here in a second. So if you're not familiar with the electron transport chain, it is a collection, literally a chain of mostly protein molecules that are sitting side by side next to each other in the inner membrane of the mitochondria. And so you don't have to memorize the names of these components, but each one of these circles here is representing a different member of that electron transport chain. And essentially the way this works is those electron carriers, the NADH and FADH molecules that we've produced in some of these other steps, are going to donate those electrons that they were carrying to this chain. So here we've got our NADH showing up at one end of the chain. It will release those electrons, so NADH would become oxidized and get converted back to NAD+. And now those electrons are going to get passed from one component to the next down the chain. And the reason they get passed along is because each component is slightly more electronegative than its neighbor. So as soon as flavin M grabs those electrons are stolen away by its neighbor, and so on down to the end of the chain. And importantly, we need oxygen, which is a very electronegative atom sitting there at the very end of the chain, and that's going to pull those electrons out of that electron transport chain. And remember, these electrons are generally being moved around in the form of hydrogens. So what we see is we pull those electrons or hydrogens out, and they combine with oxygen as we get some water being formed. But this is really the whole reason you need to breathe oxygen. Without oxygen in your cells, the electrons basically get stuck in the electron transport chain. They would just back up to the beginning of the chain. You'd have a big traffic jam, and the chain would stop working. And as I'll show you, that essentially means you're going to stop producing the majority of ATP that your cells need. So here's just a little bit more detailed cartoon of what the electron transport chain looks like. This is same idea, but it's illustrating the fact that a lot of those components of the chain are actually organized together into complexes. So we see 4 complexes here. They're numbered 1, 2, 3, and 4. And then you have these 2 components of the chain. You don't have to memorize their names, but ubiquinone and cytochrome C that are loners, so not part of any complex at all. But again, these electron carriers, NADH or FADH, are going to drop off their electrons at the top of the chain. NADH will deposit its electrons into complex 1. FADH will deposit its electrons into complex 2. But in both cases, those electrons are going to get passed down the chain again to oxygen that'll be waiting at the end to pull those out. And you can see we've got the chain mapped out on a graph here that's looking at free energy. And so what this is trying to illustrate is that as the electrons are moving down the chain, we're seeing a negative change in free energy. So energy is being released, negative delta G. So with each passage of these electrons, a little bit of energy is being released. And so you might look at this and say, well, why is there so many steps here, right? And if we go all the way back to the graph we started with in this section, right? There's been tons of steps so far, 10 reactions in glycolysis, 8 in the citric acid cycle, all these different components of the electron transport chain. If we want to get the energy out of glucose, why don't we just do it all in one big reaction? Just boom, get all that energy out at once and we're done. And the reason that doesn't work, the analogy I like to use is imagine the fuel in the gas tank of your car, right? It's analogous to the glucose fuel in your cells. So you could get the energy out of all the gas in your gas tank in one big reaction, right? You could throw a match into your gas tank and you're going to get that energy out really quickly, right? Your gas tank's going to explode. So the energy's out, but that's not very useful, right? It's not going to help you get to class or to work or wherever you need to go. And it's the same kind of idea in your cells, right? If we were to blow up glucose and get all that energy out in one step, most of that energy would just be lost in the form of heat. We couldn't really capture it to do anything useful, whereas if we can release it just a little bit at a time, right, driving the car and just stepping on the accelerator just a little bit at a time, we can do something useful with that energy. Yeah, so I think that's what I just said here. We don't want to lose that energy in the form of heat. So in the case of the electron transport chain, what are we doing with the energy that's being released from each passage of the electrons down the chain? And what the cell is doing is it's using that energy to pump protons, so positively charged hydrogen ions, across that inner membrane of the mitochondria. And so we're going to build up a bunch of these protons in what's called the inner membrane space, so the space between the outer mitochondrial membrane and the inner mitochondrial membrane. And that buildup of those protons there has a lot of potential energy. It's called proton motive force, and we're going to use that to actually make our ATP. And just to remind you, where all this is happening, right? So here's our mitochondria. We've got the smooth outer membrane. We've got inner membrane space, and then we have the folded inner membrane. Okay? And then the matrix is the yellow in the middle here. So remember, glycolysis was happening out here in the cytoplasm. Pyruvate oxidation and citric acid cycle are happening in here in the matrix. And the electron transport chain is actually embedded in this folded inner membrane. So all those complexes that I was just showing you are here in this membrane. And that's the whole reason this membrane is folded, is you want a big surface area so you can have lots of electron transport chains. So imagine that chain just being repeated over and over and over again, just packed into the surface of this folded membrane. And so what I was just saying is we're going to use that energy of the electron transport to pump protons into this space here between the membranes, so this space up here. So we're just going to build up all these protons between the two membranes. And then by this process called chemiosmosis, we're going to allow those protons to basically diffuse back across the inner membrane into the matrix, and that's where we're going to make our ATP. So if this all sounds a little bit abstract, I'll show you the cartoon here of what's going on. All right, so this looks busy, but it's really not too super complicated. So this is our mitochondria. Here's the smooth outer membrane. Here's the folded inner membrane. And then we have the inner membrane space here. And then the matrix would be down here in yellow again. And now we can see our complexes that are part of the electron transport chain here embedded in that inner mitochondrial membrane. And so all these electron carriers, most of which were being generated down here in the matrix by the citric acid cycle, right? They're going to show up and drop off their electrons either at complex 1 or complex 2, depending on whether we're talking about NADH or FADH. The electrons get passed down that chain, releasing a little bit of energy with each passage, and that energy is being used to pump these protons, the little gray circles here, across that inner membrane, so from the matrix into the intermembrane space. So we're just building up this big accumulation of hydrogen ions here, or protons, whatever you want to call them. And here's our oxygen right at the end of the electron transport chain, pulling those electrons out, preventing the traffic jam. Produce water as a byproduct there. Okay, so we've built up this big concentration of protons, and those protons don't want to be there, right? They're all positively charged. Like charges repel each other, right? They want to get away from each other. So they would love to either get out of the mitochondria or get back into the matrix. And what your cell does, what the mitochondria does, is provides it a path to do just that, to get out of the inner membrane space back to the matrix. And that path is in the form of this enzyme that's called ATP synthase. So guess what ATP synthase is going to do, right? It's going to synthesize a bunch of ATP. And this ATP synthase enzyme has literally small channels through the membrane that allow those protons to move through. And as the protons pass through these channels, they cause a shape change in part of this enzyme, and that shape change is going to allow this section of the enzyme down here to basically grab ADP, grab a phosphate, and mash them together, right, to produce ATP. So that's a reaction that requires energy, and that energy is really coming from this flow of protons, right? So we need that proton gradient to make all that ATP. And just to focus in here quickly on just a little bit more detail on the structure of ATP synthase, that enzyme. So it's got two subunits, the F0 subunit, which is the part that spans the inner mitochondrial membrane and actually has these channels for the protons to pass through. And then we have the F1 subunit, which is hanging off down here in the matrix of the mitochondria. That's the catalytic, really the enzymatic subunit that's going to produce the ATP. Okay, so again, this is called oxidative phosphorylation. Let me just go back a couple of slides here. Because we are basically phosphorylating, we're adding this phosphate group to ADP, right, to make ATP. That's the phosphorylation part in the presence of oxygen, right? We needed oxygen here to keep this chain flowing, to keep those protons pumping. So oxidative phosphorylation. All right, I've got a couple of videos you can watch if I totally lost you here. There's kind of a textbook video on Canvas that walks you through all these different steps of cellular respiration. I think it's definitely useful to take a look at. The second one is just a YouTube video that's just kind of fun. It's made by these two guys that were biology majors or biochemists at Stanford, and they put together a little song that kind of describes the whole process. That video is called Oxidate It or Love It if you want to check it out. But I'm going to stop there and we'll finish this up in part 4.